In this video, we're going to be looking at the process of protein synthesis, which we can actually break down into two kind of smaller steps known as transcription and translation. Remember that the word synthesis means to make. So this lesson is going to talk about how cells make proteins. So remember that the process of protein synthesis can be broken down into these kind of two smaller sub-steps. The first one is transcription, and the second one is known as translation. And you can see in this image here that during transcription, a molecule of DNA gets converted into a molecule of RNA. And more specifically, we're going to be talking about a type of RNA called mRNA. And that M stands for messenger. During translation, the RNA is going to be kind of converted into a protein, or at least it's going to provide the instructions needed in order to make a protein happen. Recall that genes are just sections of DNA and chromosomes that code for proteins. So you can kind of think of the genes as being the recipes for how to make proteins. In fact, one gene provides the information needed to make one protein. So your genetics or your genes determine what proteins your body makes. Remember that your genes are just these little sections of DNA that are stored in your cells in the form of these chromosomes here. Now you can see over here, this is something called a karyotype. And this is showing all of the chromosomes in a particular individual. And you can kind of see these bands on the chromosomes here. Now, each one of those bands actually can hold hundreds to thousands of genes. So each band is not its own gene. The genes are even smaller than that. But genes can be made up of a really long strand of DNA in order to provide that information needed so your body can make a protein. So let's review a little bit about where DNA is located, what organelles we need in order for protein synthesis to occur, and where the organelles for protein synthesis are located. So remember that in eukaryotes, one kind of defining feature of eukaryote is a cell that has a nucleus. All right, and this is a membrane-bound nucleus. So it's a nucleus surrounded by a membrane, kind of like the cell membrane. So the answer to this question is that DNA is located in this membrane-bound nucleus. Now, the organelle that's necessary for protein synthesis to occur are the ribosomes. And remember that the ribosomes are going to be located in the cytoplasm, or you could say the cytosol. Now, they may be free-floating in the cytoplasm, or they may be attached to the rough ER, which is the rough endoplasmic reticulum. Even if they're attached to the rough ER, they're still located in the cytoplasm. So the key thing is that we have to get the information from the nucleus to the cytoplasm. And that's where our RNA is going to come into play. So as we outlined on the last slide, we need DNA to make proteins, but DNA is stuck in the nucleus and the ribosomes are stuck in the cytoplasm. So how do we get the information from the nucleus to the cytoplasm? Because DNA is too big to leave the nucleus. So how do we get this information stored in DNA from the nucleus out into the cytoplasm. And the answer to that conundrum is this molecule known as mRNA. Now, mRNA is smaller than DNA. And it's smaller in two ways. So it is shorter than DNA. It's also only a single strand. And because of its size, it is able to fit through the pores in the nuclear membrane. So mRNA is made in the nucleus, but then it is able to leave the nucleus through these little nuclear pores, which are just little openings in the nuclear membrane. And then it can go out into the cytoplasm to find a ribosome. During the first part of protein synthesis, known as transcription, a molecule of mRNA is made from a molecule of DNA. So a gene, which is, of course, a segment of DNA, gets converted into a molecule of mRNA. Now, this conversion happens through complementary base pairing, which is similar to what you learned when you learned about DNA replication. Remember that during DNA replication, you have these two strands of DNA that are complementary to each other. So you've got your G, C, A, and T. And then the complementary strand, we know that C always pairs with G pairs with C pairs with G, A pairs with T, and T pairs with A. Now during DNA replication, these two strands are going to separate. And then each of the original strands becomes like a template for a new strand that's going to be made. And the new strand is just complementary to the old strand. All right, and I'm out of room, but you should remember that from when we did DNA replication. Now, in transcription, something kind of similar happens, but instead of the new strand being DNA, instead that new strand is going to be RNA. And the two strands of DNA are not separating completely, and they're not separating forever. It's actually just a temporary separation of the two strands of DNA, and it's only a portion that gets separated. So what happens is that you have this double helix of DNA, and what happens is that just a section of it is going to open up. And this is the section that is the gene that is being expressed. So this DNA kind of opens up. And then we have enzymes known as RNA polymerase that is going to come in and basically make a strand of mRNA using the DNA as a template. So this area in yellow here is going to be our mRNA. Now, another key difference is that RNA is made up of nucleotides, just like DNA is. So remember that nucleotides are made up of a 5-carbon sugar, a phosphate group, and a base. Now, the 5-carbon sugar in DNA is known as deoxyribose, but in RNA, it's known as ribose. So it's ribose in RNA, and it's deoxyribose in DNA. The other key difference has to do with the types of bases that are present. So in DNA, we have what we call, or what we kind of remember as GCAT, where guanine always pairs with cytosine, and adenine always pairs with thymine. Now, mRNA does not have thymine. Instead, it has what I like to call GCow. So instead of thymine, it has this molecule known as uracil. So no thymine. Instead, it has uracil. And the base pairing still applies, where G still pairs with C, and A pairs, in this case, with U instead of T. So if we have a molecule of DNA with the bases of GCAT, the corresponding RNA, G is still going to pair with C, and C is going to pair with G. Now, if this were DNA over here, which is not, instead it's RNA, there would be a T here. But remember, we don't have T. So instead, that A is going to pair with a uracil. Now, the T is still going to pair up with A because we still have adenine. So really, the only difference is going to be that U pairs up with A. With the U is in RNA, and it's going to pair up with the adenine in DNA. So to recap, some differences between DNA and RNA. So first of all, mRNA stands for messenger ribonucleic acid. So the M stands for messenger, and then ribonucleic acid. And we can break down this word a little bit. So ribo refers to the ribose sugar, which is just a 5-carbon sugar. And so it's going to look kind of like a little ring, kind of like a little pentagon. The nucleic acid part of it tells you that it is a nucleic acid, which remember is one of our macromolecules. And remember that macromolecules like DNA and RNA, like our nucleic acids, are made up of monomers, which are the nitrogenous bases. And then remember the big difference between RNA and DNA is this base known as uracil. So in DNA, this base would be thymine. But in RNA, it's called uracil. The other main difference, as you can see in the image over here, is that DNA is double-stranded, whereas RNA is single-stranded. So this makes RNA thinner, and it's one reason why it can leave the nucleus and go out into the cytoplasm, is because it can fit through those small pores in the nuclear membrane.